My name is Andreea Avramescu. I'm currently in the second year of my PhD at the Business School of the University of Manchester. The paper I'm going to present today is the result of a part of that research conducted together with Dr. Richard Allmendinger and Dr. Manuel López-Ibáñez. The title of the presentation and automatically of the paper is a multi-objective, multi-type facility location problem for the delivery of personalized medicine. Accordingly, my presentation's main topics of discussion will be on supply chain optimization with focus on the personalized medical products. So firstly, have you ever wondered why some medication for like migraines or other headaches never worked for you, even though they are very well-known treatments for those symptoms? Well, there isn't really anything unusual behind it. It is widely accepted nowadays that the traditional pharmaceuticals are effective for only about 60% of the population. This is because our reaction to a drug is influenced by factors such as our diet, environmental factors, other health conditions, or maybe other drugs we are currently taking, or simply the aging process. The medical research then moved towards understanding these differences, and we now know that our organism's response to drugs is driven by genetic, environmental, and social conditions. All this led to an increased interest in understanding patient variability and discussing completely new treatments and approaches under an umbrella term of personalized medicine. The biopharmaceutical products that have been created as a result of the development of personalized medicines are known as advanced therapies medicinal products, or simply ATMPs. ATMPs can treat progressive diseases such as cancer and rare disorders in most cases where there is no other treatment available. However, the delivery of these products brings challenges which cannot be solved using the existing supply chain configurations. To name a few of these characteristics, they have a lengthy manufacturing process that works with a high level of certification and low global demand. They also have a very short shelf life of only a few days. Thus, now we are shifting from a continuous off-the-shelf and mass production of traditional drugs to on-demand and batch mode of production for personalized therapies. As a direct consequence, these treatments are the most expensive medical therapies available currently, with prices up to $2.1 million. For these reasons, we only have about 80 such products available with commercial authorization and are only used as a last resort. Currently, this is one of the fastest progressing areas of pharmaceutical industry, with more than 2,000 products in clinical trials. It thus becomes of great importance to create network configurations capable to support the biopharmaceutical companies with the manufacturing and delivery of these products. This brings us to the ATMPs network. I'll very, very briefly describe their vein-to-vein supply chain without going into too many medical details. Most commonly, these products are divided into two categories. The autologous, meaning that the patient is also the donor, and allogeneic, where the donor and the patient are different individuals. Our focus is on the autologous products, and so I will only discuss this supply chain. In this case, the supply chain starts with the patient, whose cells are collected at the hospital through a process called leukapheresis. It simply means that the blood is separated into individual components. The resulting product is time and temperature sensitive and needs to be transported either fresh or frozen to a manufacturing site where the cells will be genetically modified. After the entire manufacturing process is completed, the modified cells will be returned to the exact same patient with the ability to treat or even cure the disease. A bit more formally speaking, we have a set of candidate locations, and for each candidate location, we need to decide if we open a manufacturing facility or a cryopreservation facility, or maybe nothing at all. We also have a hot set of hospitals whose location we already know, with an associated demand of orders where each patient has one order, and for each hospital, we need to decide which manufacturing facility it is assigned to, and if the order is frozen, which cryopreservation facility. As previously mentioned, our supply chain starts and ends with the patient, so the ATMPs must be returned to the same hospital from where the starting material was collected. We then have two possible scenarios. The collected product is not cryopreserved, then our network goes from the hospital to the manufacturing facility and then back to the hospital. However, if the process from hospital collection until the cells are returned to the patient is longer than the shelf life, then our network goes from hospital to an independent cryopreservation facility, to a manufacturing facility, and then finally back to the hospital. Our aim is then to find optimal locations for the manufacturing and cryopreservation facilities. In our case, the decision maker is a private biopharmaceutical company. We model the problem as multi-objective. Objective 1 minimizes the average delivery time per patient. As each order corresponds to one patient and has hospitals as a starting point, the objective actually minimizes the travel time from a hospital to its assigned manufacturing facility and then back to the hospital, possibly via a cryopreservation facility if it is frozen. The second objective minimizes the total cost of the supply chain. This was calculated as the cost of opening, manufacturing, and cryopreservation facilities, cost that is independent of the number of orders assigned to them, and the cost of operating the facilities, which depends on the number of orders assigned. Finally, our last objective maximizes the number of patients that can receive the therapy. This actually maximizes the number of orders that are assigned to a manufacturing facility, since that's where the orders are processed. Then the most important constraints in the ATMP supply chains are related to time constraints. So we need to make sure that the order is delivered within its shelf life. We also need to ensure that we have at least one manufacturing facility placed, otherwise the orders cannot be processed. An order cannot be assigned to more than one manufacturing facility, and only if frozen, one cryopreservation facility. Finally, we restrict that no more than one facility can be placed at each location, because manufacturing facilities already have an integrated cryopreservation unit. So it would be redundant to have both types of facilities at the same location. We test our proposed model using publicly available data of a personalized medicine product, Kymriah, manufactured and distributed by the biopharmaceutical company Novartis. Kymriah is a genetically engineered cell therapy that first obtained market authorization in August 2017, and automatically the breakthrough therapy designation of personalized medicine. While the product has recently obtained authorization to expand their targeted patients, the demand data was calculated using only their first designation, which was for pediatric patients suffering of leukemia. The data was obtained from the International Incidence of Childhood Cancer and supplemented with some expert opinions from the literature. Because of these differences and some other challenges and differences in the modes of production, we did not attempt in this paper to compare our results with the current Kymriah network and their current cost and delivery time. Moving to the location data, as I said, we already know the location of the hospitals. For a hospital to offer such a therapy, it needs to have FACT accreditation, which is cell and gene therapy accreditation. So we used all those hospitals in the USA. Then for the candidate locations for both manufacturing and cryopreservation, we had the 1,000 biggest USA cities and calculated the distance between any two locations using hovers in this function. Now talking about the manufacturing data. The data used to calculate the total duration and cost of the manufacturing process, which is described here in a bit more detail, was provided by Biopharm Services. Biopharm is a consultancy company working with biopharmaceutical companies, offering support with different supply chain concerns. We also work closely with the company to ensure that our models and the data we use are applied and tested on real-world scenarios. This flowchart on the right side shows a simplified version of the different steps of the delivery and manufacturing processes together with the necessary resources at each step. Perhaps it is worth mentioning now that the data to calculate the cost is simplified. We did not consider the cost of raw materials or other indirect costs, which are usually added to the total price of an ATMP, but this will all be considered in future research. So the above problem, we use the NHJ2 algorithm and the multi-objective random search as baseline. The NHJ2 is a multi-objective genetic algorithm, and given today's audience, I won't explain it into great detail, but I want to mention its three main features. Namely, it uses non-dominated sorting, through which all individuals are sorted according to the level of non-domination. This means that a solution A dominates a solution B if and only if all objectives of solution A are not worse than those of solution B, and at least one of the objectives is better. Secondly, the algorithm is elitist, as only the most fit individuals are guaranteed a place in the next generation. And finally, the third feature is crowding distance, which forces the algorithm to emphasize on less crowded solutions to ensure diversity. To initialize the population, for both algorithms, we used a novel procedure, which we called progressive coverage, and later compared it with random population initialization. To briefly describe our progressive coverage algorithm, basically we used a prior knowledge of the shelf life and the radius of each facility to find the minimum number of cryopreservation facilities that can cover the demand. It is important here to note that since an order cannot be processed without a manufacturing facility, we prioritized this type of facility and started the solution with it, and then placed the cryopreservation around it. We then use an iterative approach and look something like this. So looking at all orders that are not within the radius of any placed manufacturing facility that was placed here, the cryopreservation facility is placed at the position that covers most of the uncovered hospitals. If several candidate locations cover an equal number of uncovered orders, then the location with the maximum number of potentially assigned orders would be selected. This approach was preferred in order to increase the resilience of the supply chain in case of disruptions of one of the manufacturing facilities. The process then is repeated until the entire demand is covered and all the solutions are added to the initial population. This process is also repeated until the population size is reached. By replacing the random initialization with this approach, the algorithms were found to find better solutions in less iterations. And this we found by looking at the hypervolume. So this figure over here shows the average hypervolume and the corresponding 95% confidence interval. It is apparent that the NSGA2 manages to achieve significantly higher hypervolumes than the random search when compared with the same initialization procedure. And down here is the one with the random initialization, and this one has the progressive coverage that I just explained. However, the algorithms using the progressive coverage perform better overall than those applying to the random one, which indicates us how important this initialization is for our problem. A solution in our case then includes the number of manufacturing and cryopreservation facilities, their corresponding locations, and the values of the three objective functions, namely the cost of the supply chain, the average waiting time, and how many orders are covered. And here is one such example. We decided to show a possible extension that covers the entire demand. So we've got the 100% coverage, but also using the current manufacturing facility of Kimbria, which is over here in Morris Plains, New Jersey. The solution has overall 6 manufacturing facilities, which are shown with these purple map placeholders, and 2 cryopreservation facilities with the green map placeholders. The circles correspond to the radiuses of each facility type. So the same purple will be for manufacturing and green will be for cryopreservation. Some general notes, for example, given the higher number of hospitals on the eastern half of the USA, there were considerably more manufacturing facilities placed here compared to the western side. This distribution leads to a lower average delivery time, but the overlapping of manufacturing radiuses, for example, here, indicates that it's possible to reduce the cost by gradually increasing the time objective. However, because we assume that the orders are assigned only from existing hospitals, there are small areas of the USA still uncovered, like these two up here, for example. Since there is a possibility for the hospitals, for other hospitals to obtain that accreditation that I mentioned earlier, and so start to offer these personalized therapies, the demand coverage would be reduced. In terms of resiliency, this configuration has a high backup coverage in case of possible disruptions to one of the production sites, as all hospitals are within the shelf life radius of at least one of the cryopreservation facility. However, given the unbalanced distribution of hospitals between the 2 facilities, one of them might become overloaded and the solution unfeasible should we consider capacity constraints in future work. This slide presents some overall interesting results. For example, up here, it shows that an increase in the orders leads to an increase in the cost, but has no direct impact on the delivery time. This is because coverage can be achieved either with cheap cryopreservation or a bit more expensive manufacturing facilities. The former leads to a higher delivery time, while the latter shortens that delivery time. Thus, there is no direct relationship between coverage and delivery time. A higher number of manufacturing facilities in a solution corresponds to a lower average delivery time, higher costs, and higher coverage, as I mentioned earlier. We did not include the full Pareto fronts in this presentation, but we compared the NSGA2 with progressive coverage and random search without it. So basically our two extremes. What is interesting in this was that the first is placing more manufacturing facilities, while the second one is placing more cryopreservation facilities overall. In this, it leads for the second one to scenarios including solutions with better cost, but almost no coverage and a high average delivery time. We, however, included two Paretos. Both of these correspond to the algorithms using progressive coverage. The solutions in blue are the solutions of the NSGA2, and the ones in yellow are the ones for random search. The axis corresponds to the three objectives. So you have the average waiting time, cost, and coverage. To make the plot easier to visualize, we inverse the coverage, and close to 1 up here means that there are almost no orders covered, while 0 means that there is a perfect coverage. While the two figures might look almost identical, we then run the non-domination on all solutions, and this is the final Pareto, and it is clear that almost entirely the solutions of the random search are dominated by those for the NSGA2, especially that the solutions we really aim for are the ones close to the origin, because we want solutions with low delivery time, low cost, and to cover as many orders as possible. So in conclusion, I argue today that the traditional pharmaceuticals are being challenged by the people's ability to create more personalized breakthrough therapies. These new treatments are creating possibilities to treat life-endangering diseases for which no other treatment is feasible or currently available. However, they are complex living products, and the existing research in logistics and supply management does not provide sufficient knowledge that can sustain their commercialization. We have presented a model dealing with the location-allocation of the manufacturing and cryopreservation facilities, capturing the trade-offs of cost between cost, delivery time, and demand coverage. And we also used a new initialisation procedure which was found to be superior when used for both multi-objective genetic and random search algorithm. Broadly translated, our findings indicate that the operations research field has the potential to help biopharmaceutical companies offer timely and cost-efficient products, thus making revolutionary therapies more widely available. But there remains the question if this is it. But we as researchers and patients are just starting to really understand personalized medicine and its implications, and there is so much more that can and should be researched. The current study can be continued in different ways. For instance, we did not try to optimize the manufacturing process. To create a more general landscape of biopharmaceutical products, we need to consider multiple products type. The optimization of the manufacturing steps will also be really important. Our case study is also just the first step, but expansion to global demand will include a higher number of constraints, such as regulation procedure and certain approvals, while the shelf life restrictions will become even more challenging. Because of this, we can use various cryopreservation methods, and additional storage facilities could also be used. However, the cryopreservation procedure needs to have a thawing after it, and that thawing can increase the risk of damaging the material. Maintaining the cells viable is a crucial component, because we work with patients with advanced stage diseases and poor health condition might preclude running another apheresis procedure. Thus, we should also consider scheduling and patient prioritization based on risk and potential success rate should also be essential. Okay, thank you for listening. And if you have any comments or questions, I will try my best to address them.
That's great. Super talk. Thanks very much, and kept brilliantly to time as well. Can I ask if anybody's got any questions? Please, we're quite a small group, so please just unmute your microphone and ask away. Well, while people are thinking of questions, one I have is, I'm really interested in the practical application of this. How does, are you aware of any pharma companies or companies using machine tools at the moment, or are they using traditional statistical approaches? And have you had any interest in your work?
Okay, that's a question I could probably talk for hours about. So not personally, we are not working directly with the biopharmaceutical companies, but we are working with a consultancy company that works directly with the biopharmaceutical companies. Yep. So we are always trying to understand what their current approach is. Currently, they're most focused on developing the biopharmaceutical and trying to optimize the manufacturing process by itself, which ended up not being an efficient way, because it led to really big challenges for companies that had to discontinue their products or had to deliver lower quality treatments. So they're using, I think AI is probably one of the famous they're using to solve the problem, but I wouldn't know exactly which options, but they're definitely looking at optimization, and the supply chain is starting to play an incredible role now. That there are only 4 papers published, and all of them were in the past 2 years on this exact topic.
Oh, I mean, it's really interesting, because I could imagine if you can save, I mean, it's probably worth a lot of money to the company to be able to be more efficient, I'm sure. Because,
it is, and it ultimately, it's not about whether the product is cheaper, because even if we don't consider that, it ends up with being, is it cost efficient and is it worth paying for it? Because when you talk about products that are up to $2 million to get approval for market to actually sell your product, then you don't really want to have that kind of expensive product, because nobody will reimburse the patient for it and nobody will buy your product. So it's definitely, you want to have as many cost-efficient products as possible.
Good. And one question that's probably naive, but I'm dying to ask it, is do you think that some of your work could have been applied to the rollout of the COVID vaccine as well, in being able to ensure better, especially with the Pfizer one, which has obviously quite strict handling conditions?
We actually discussed it, and yes, our supply chain stays somewhere at the intersection between multiple supply chains, and vaccine supply chain is actually one of them, and it shares lots of common characteristics with it. So it would definitely be a nice extension, and it could be applied to it. But actually, biopharmaceutical companies themselves had some problems during COVID with all the restrictions between countries. So, but yes, it would definitely be could be useful to apply.
That's great. Okay. Anybody have any other questions?
Yes, I actually have one. First of all, the talk was really interesting. When you evaluate your cost function, you mentioned that you are not considering of course, some of the possible costs, because it's really hard to take everything into account. And in your opinion, is this part realistic? Like, is this ignoring this cost? Is it just a small percentage, or like, do you have an evaluation of that?
It is a realistic scenario to evaluate the cost for the delivery of the product. It becomes more complicated. So I can say that our cost does match the real world scenario of delivery, but not the manufacturing processes. The manufacturing process, it's really expensive in itself. It includes lots of different variables, and it includes lots of different costs, and it's really hard to actually build that model. And we're working on it. We'll hope to actually have the model including the cost for manufacturing as well at the end. But this one we created, it's realistic for delivery. It is not realistic for the entire cost of the supply chain.
All right, thanks.
Brilliant, thank you. Any final questions for Andreea? Fantastic. Okay, well, thanks again for a brilliant talk. That's really enjoyable. Thank you.